Let's talk about mutations.
Well, what are mutations?
They are changes in the genetic material in a cell or virus, usually caused by mutagens.
Oh yeah, aren't there 3 types of mutations?
Well, there are multiple types of mutations, but 3 types of point mutations that I want to talk about now.
Well, what are they?
Base pair substitution and base pair insertion or deletion. Now that we got that out of the way, let's go into detail. In base pair substitution mutation, one nucleotide pair replaces another nucleotide pair. A missense mutation still codes for amino acid, but not quite the right one. Whereas a nonsense mutation will change an amino acid codon to a stop codon, most likely making a non-functional protein. In base pair insertion, a nucleotide pair in a gene is added. In deletion, a nucleotide pair is deleted. When this occurs, a frameshift mutation usually happens.
Now that's a lot of info.
Yeah, and that's not all. Let's look at how mutations affect mitosis in the cell cycle. We start with the diploid somatic cell. Interphase occurs. Now we have a cell with 2 complete sets of chromosomes. That cell splits. Now we have 2 genetically identical daughter cells. Now to look at mutations, we're going to zoom in.
There are 2 types of regulatory proteins involved in cell cycle control, cyclins and CDKs.
NPF is a cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into M phase of the cycle. Mutations in these proteins can cause oncogenes. Proto-oncogenes are normal cellular genes that are responsible for normal cell growth and division. However, they can be converted to oncogenes. Amplification of a proto-oncogene increases the number of copies of the gene, whereas point mutation causes an increase in gene expression. Mutations in the RAS proto-oncogene and p53 tumor suppressor gene are common in human cancers. When there are mutations in the RAS proto-oncogene, there can be a production of a hyperactive RAS protein and increase in cell division. In the case of damage to a cell, p53 is important. When there is a mutation in p53, suppression of the cell cycle may be prevented. Well, what happens in meiosis? A common type of mutation in meiosis is non-disjunction. In non-disjunction, pairs of homologous chromosomes do not separate normally during meiosis. As a result, one gamete receives 2 of the same type of chromosomes and another gamete receives no copy. When a zygote only has 1 copy of a particular chromosome, it's called monosomic. When it has 3, it's called trisomic. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred.